Hello everyone, this is Nalini from Maharani Science College for Women, Department of Botany. In this session, I will discuss about the mechanism of enzyme action. How exactly the enzymes work, the mode of action of the enzymes to convert the substrates into products. We all know most of the chemical reactions, some molecules will possess higher kinetic energy when compared to the other molecules. The molecules with higher kinetic energy can easily show the conversion of the reactants into products because of their agitation, because of their collisions between the atoms. But those molecules which possess lower kinetic energy, they fail to convert the reactants into products. According to Arrhenius, this lower kinetic energy molecules are called as energy poor molecules, and higher kinetic energy molecules are energy rich molecules. So how to overcome this energy barrier, which is actually make the reactants non-functional or fails the molecules to convert into products. So this the extra energy required for the chemical reaction to complete is called activation energy. This activation energy is met in case of in vitro conditions or inside the glass. It can be overcome by increasing the temperature of the reaction mixture. Due to thermal agitation, the molecules will acquire some extra kinetic energy that will make them to proceed and convert into products. But this cannot be done always, mainly because the reactants may lose their properties because of the increase in temperature, because of the excess of heat of the reaction mixture. So how to overcome this energy barrier? Arrhenius has given some examples where the chemical substance which is proteinaceous is giving some interesting results by increasing the reactants to convert into products. How exactly this happens? This happens in vivo conditions with the involvement of the enzymes. That those enzymatic reactions where the activation energy increase is done with the help of the catalyst, that is the enzyme. So the alternative is to lower the kinetic activation energy requirement. What exactly these enzymes do? In place of increasing the temperature, this enzyme is going to lower the activation energy. The requirement of the extra kinetic energy will be lowered with the enzyme molecules because of their colloidal nature, because of their catalytic activity. They will lower the requirement of the activation energy. So usually in all the chemical reactions there will be a transition state where exactly the intermediate complex, that is enzyme substrate complex, is framed. That particular transition state that requires the lower energy in case of catalytic reactions, in non-catalytic reaction, to reach this intermediary stage or the transition state, the energy requirement is more, activation energy requirement is more. The unstable intermediate compound quickly breaks down, this transition state compounds will quickly break down to form the reaction products. And in enzymatic reactions, the enzymes are released without undergoing any change. Arrhenius first pointed out all the molecules in a given population do not have the same kinetic energy. Kinetic means the molecules will be in motion, in movement. This movement creates some collision and they have more energy by colliding with each other, and the energy rich molecules will be functional, while others or energy poor molecules, they fail to convert the reactants into products. In an ordinary chemical reaction, only energy rich molecules can take part in the reaction at normal temperature. Due to energy barrier, the lower energy molecules fail to convert into products. The higher the energy barrier for the molecule, the greater is its stability. Stability is nothing but inactiveness to take part in the reaction. So the higher the energy barrier, the molecule will be more stable or more inactive, and it cannot convert into product. The energy required to handle the molecules over this energy barrier is called activation energy. In a non-enzymatic reaction, the energy barrier can be provided by increasing the temperature of the reaction mixture. Enzymes do not increase the kinetic energy of the molecules, but they combine with the molecules and lower their requirement of the activation energy, which is necessary for the completion of reaction. As the enzymes form enzyme substrate complex, that is the transition state or intermediary complex, which overcomes the activation energy barrier and gives the products. In case of enzyme catalyzed reactions, the rate of reaction is optimum at normal temperatures. It is because all the molecules, that is energy rich and energy poor molecules, can combine with the active sites of the enzyme, and they will form the transition state or the ES complex or enzyme substrate complex, which later break down into the enzyme unchanged and the products are liberated. Increased temperature improves the kinetic energy of the molecules, that speeds up the rate of reaction. This is mainly achieved by the thermal agitation between the molecules. But in vivo conditions, inside the living organism, this will not work. This increase in temperature will always not work, because it will denature the proteins, and that will harm the individual or the microorganism or any living organism. In other words, the incident by lowering the energy of activation of the reactions. How exactly the in vivo conditions work? That is not by increasing the thermal agitation, but by lowering the activation energy requirement. In vivo conditions, enzyme action is greatly achieved by temperature. If temperature is increased at 10 degrees centigrade till certain threshold, at every increase in the 10 degrees centigrade, the chemical reaction shows the rate of chemical reaction will increase twice, double the rate. Okay, if the threshold of that optimum temperature is reached, then the enzymatic protein will get denatured and the reaction starts drooping or decrease. But at initial stages, every 10 degree increase in temperature will show the chemical reaction doubled. So this diagram shows us how exactly the enzymatic reactions will reduce the free energy requirement, or the activation energy requirement will be lowered in case of enzymatic reactions. Here we can see these two are the reactants. At this point we can see the reactants are present in the reaction mixture, and they require maximum free energy or the activation energy. So we can see this line in uncatalyzed reactions. To reach the transition state in uncatalyzed reactions, the energy requirement is very much high when compared to the enzymatic reactions where the energy requirement is very less. We can see the energy requirement is very low in case of enzymatic reactions. This transition state is reached very quickly by lowering the activation energy. That is the difference between an uncatalyzed and catalyzed reaction. So the transition rate is quickly reached and the products are obtained very quickly in case of enzymatic reactions. This is always required in vivo conditions. Let's move on to the next one. That is some of the models and has been described to explain the mode of enzyme actions. The theories supporting the enzyme substrate interactions. These can be called as models or theories supporting the enzyme substrate interactions. First one, the very famous lock and key hypothesis, which is proposed by the scientist Fisher. So it is popularly called as Fisher's lock and key hypothesis. In this, as only a specific key fits to the particular lock to open. Similarly, a specific substrate should come and bind to the active site of the specific enzyme to give the enzyme substrate complex, that is the transition state, to be formed. And later it breaks down the substrates into the products and enzyme is set free. According to Fisher, classical locking key theory or lock and key theory, which is widely accepted, that there is a physical contact between inside and the substrate. There will be an interaction of the enzyme with the substrate. They will go deep into each other and they are going to make a enzyme substrate complex. This theory was supported from the study of the competitive inhibition of the enzyme activity. How exactly the competitive inhibitors, which resemble in the structure as that of the substrates, how they are going to block the active sites before the substrates come and bind. The competitive inhibitors have some structural similarity with the substrate molecule, both of which compete for the same active site on an enzyme. Let's see how exactly the particular key is going to get fit into a particular lock. In the same way, the key will be the substrate and lock will be the enzyme here, and they should fit into each other. Then only the enzyme substrate complex or the transition state or intermediate state will be reached, which will facilitate in the formation of products later. If even some part of the active site is preoccupied by any of the structurally similar competitive inhibitor, then the substrate will not be able to combine with the active site of the enzyme, and thus the wrong key cannot fit into the lock. Thus no products are liberated. Removal of these competitive inhibitor clears the way for the substrate to come and bind to the active site, and thus there is a formation of transition state or the enzyme substrate complex, which later forms the products. So we can see one more diagrammatic representation of the phenomena, Fisher's phenomena of lock and key mechanism. See, very important here is the formation of this enzyme substrate complex. We can see enzymes which are designed like that. They have some grooves those grooves are very much important. They are called as active sites, which will make the exact combination of the substrate, which is having the same structural phenomena or the conformation, which will help in binding the substrate to the enzymatic grooves and making the transition state or enzyme substrate complex. So later what happens, the conversion of this the ESR, enzyme substrate complex, into a suitable product, and enzyme is released unchanged. So this is the popular lock and key mechanism proposed by the scientist Fisher. Then the next model or next theory supporting the enzymatic activity or the mode of enzyme action is Koshland's induced fit theory. As the name itself is telling, there will be some changes or induction or changes in the conformation of the enzyme or in the structure of the enzyme will be induced because of attachment of the substrates to a particular enzyme. So this is Koshland's induced fit theory. Let us see how it works. According to this theory, when a suitable substrate approaches the active site of the enzyme and it is going to frame the enzyme substrate complex, the substrate actually induces the conformational change in the enzyme. As a result, there will be some attractive groups and buttressing groups. Very important here. How exactly, according to Koshland, how exactly the enzyme molecule will be? The enzyme molecule will be having two important sites. This arm of the enzyme will be having the buttressing site or attachment site. This site will facilitate the substrates to come and bind to this particular arm of the enzyme. Here it is another arm of the enzyme, that is the catalytic site. This catalytic site will be helping in the breaking of the older bonds and making the new bonds. The catalytic activity is actually seen in this particular arm. Whenever the enzyme is having some suitable substrates, when the substrates approaches the enzyme, they will first get attracted to the buttressing site and they will bind to this buttressing site, and the buttressing site is going to help in the activity of the enzyme, and the catalytic site is going to help. Catalytic site will come close to the buttressing site so that they can undertake the action with the reactants. So that is how the conformational change in the enzyme takes place. This particular arm of the enzyme will bend and come close to the substrates, that is come closer to the buttressing site, and they will frame the bonds and easily convert the substrates into products. And finally the enzyme will be released in the original state. Why? Because the enzymes will be having the buttressing site will not be having any affinity with the products. They will help only in the binding of the substrate. The products are not going to bind to the buttressing site. So once again the conformation of the enzyme molecule will be it will come back to the original conformation, original structure, and thus the Koshland's induced fit theory works. Excuse me. The strain which weakens the bond, which ultimately broken down and helps the enzymes to convert into products. So one thing is very much noticeable here in both lock and key mechanism and also in Koshland's induced fit theory. the formation of the enzyme substrate complex is same. We can see one more diagrammatic example here, how exactly the substrates come and bind to the active site of the enzyme, making the enzyme substrate complex that will work. The substrates will be attaching to the enzyme, and enzyme will show the breakdown of the older bonds and making the new bonds with the help of the catalytic site. But Retzing site is the site which is making the attachment, making way for the attachment of the substrates, and catalytic site will make the new bonds by breaking the older bonds in the substrate molecules and release the products. And finally, the catalytic site will come back to its original position, and the enzyme is released without undergoing any chemical change. So this is the mode of action of the enzymes, how exactly enzymes act. So the summary of this topic tells us how important to overcome the energy barrier, that is some energy poor molecules or the molecules with the lower kinetic energy should overcome this energy barrier that is achieved in vivo conditions with the help of accompanying of the enzyme with the substrate molecules that will lower the activation energy requirement, rather increase in the temperature as we see in the in vitro conditions, that helps in gaining the molecules extra energy by the thermal agitation. That is achieved here with the help of the enzyme molecule, and that will make the lowering of the requirement of the activation energy and convert the substrates into the transition state or enzyme substrate complex, and later the enzymatic activity will be done with the release of products, and enzyme will be set free without undergoing any change. This is the lock and key phenomena, and also in Koshland's induced fit theory they are same, but rather they are having the induction of the conformational change in the Koshland's theory, but the conformation is already set, that is the active sites are already present in case of lock and key phenomena. But in both, the formation of the transition state or enzyme substrate complex is unique and it is same. This completes the mechanism of enzyme action. The most familiar question will be explain the mode of enzyme action with proper theories or with proper examples, or sometimes they can ask explain the lock and key phenomena. Then we need to explain the mode of action with respect to the Fisher's lock and key phenomena. Likewise, you will get 5 marks question and 10 marks questions from this topic. These are few references. You can go through these websites for better understanding. Happy learning. Thank you.